The majority of the drugs that are available to us are actually going to be antibiotics. However, there are drugs that are antifungals and antivirals, so we're going to take a quick look at those. Antifungal drugs are interesting because fungi are eukaryotic, just like human cells, so they use the same mechanisms to synthesize proteins and nucleic acids as we do. So it's difficult to find something that will kill fungal cells and not human cells. As I mentioned earlier, antifungals will target sterols because the sterol in the fungal membrane is different than the sterol in the human membrane. Antifungals can also inhibit cell wall synthesis. This is effective because fungus do have cell walls while human and animals do not have cell walls. They can also inhibit the synthesis of RNA. Only fungal cells can convert this flu cytosine into a compound that can be incorporated into the RNA, and then if it's incorporated, it will eventually disrupt protein synthesis. Human cells cannot incorporate flu cytosine, and so it doesn't have any effect on our cells whatsoever. 99% of the population 90% of the U.S. population suffers from viral diseases each year. In fact, viral diseases in the U.S. are much more common than bacterial diseases. However, there are few antiviral drugs available relative to antibacterial drugs because viruses replicate within the host cells using our own genetic and metabolic mechanisms. So it's difficult to target a virus without damaging the host. However, there are 3 methods that can be used for antiviral drugs. First of all, nucleoside and nucleotide analogs, they use a method of inhibiting nucleic acid synthesis and thereby shut down the system before protein synthesis can even begin. These are competitive inhibitors. They resemble nucleosides, and when they are used as a substrate instead of the real nucleoside, it'll cause a mutation rate in the viral RNA to increase until the accumulation of all these mutations will actually kill the virus. And it works because the virus-infected cells process nucleosides differently than non-infected cells. So remember, the virus uses the host cell's mechanisms to replicate. However, an advantage to us is that our cells that are infected with viruses will replicate just a little bit differently. They can also be enzyme inhibitors, as we saw with other competitive inhibition, and they can also be interferons. Viruses, we talked about how when they infect cells, those cells often can naturally produce what we call interferons, which are chemical signals that alert other host cells to the virus and say, hey guys, there's a virus here, watch out, and those cells can then kind of be alerted to that and prevent it, so it will inhibit the spread of infections. There are some antiviral drugs that can be given that will actually cause your own cells to increase the interferons that are produced. Protozoan and helminthic diseases are really difficult to treat because they are eukaryotic, and therefore their cells are structurally and functionally similar to human cells. However, there are a few drugs that have been used, in fact have been used for a long time. Chloroquine is a synthetic derivative of quinine, and quinine has been used for the protozoa that cause malaria since before the 1600s. Mebendazole will inhibit nutrient absorption of intestinal worms, but that is really the only, well, not the only one, but it's one of the few antiprotozoan or antihelminthic drugs that we actually understand the mode of action for. We don't really know how a lot of these other antiprotozoan or antihelminthic drugs work. But a lot of them have only been discovered in the last 40 years. The book doesn't really talk too much about these drugs, so we're just gonna kind of mention them here. But do know how to recognize chloroquine, mebendazole, and ivermectin, which I can't pronounce those correctly, but be able to recognize those as being antiprotozoan and antihelminthic. Different microbe strains have different degrees of susceptibility to antimicrobial drugs, and also the susceptibility of microorganisms can change over time. So it's good to know the sensitivities of the pathogens before an appropriate treatment is started. So tests that we can use to know the sensitivities of pathogens can define what we call the MIC, or the minimal inhibitory concentration. This is going to be the lowest antibiotic concentration that is effective. The other thing that they can tell us is the MBC, which is the minimal bactericidal concentration. These are going to be the lowest bactericidal concentration that is effective, obviously. Knowing these two pieces of information will prevent excessive or incorrect use of expensive antibiotics, and also minimize the chance of toxic reactions to larger than necessary doses. I'm going to go ahead and pause here, and we'll start back up here on section 4.